So here we are, and now we talk about large insert vectors or artificial chromosomes. For the rest of the talk, we're going to discuss two types of artificial chromosomes. One is the bacterial artificial chromosome, and the other is the yeast artificial chromosome. We now begin with bacterial artificial chromosomes first. So bacterial artificial chromosomes are engineered F plasmids or fertility plasmid. The cells that contain a fertility plasmid are called as F plus plasmids. The cells that do not have a fertility plasmid are known as F minus plasmids. And conjugation can happen only between cells that is an F plus versus a cell that is an F minus, right? So therefore, the fertility plasmid or F plasmid facilitates exchange of genetic material via conjugation in strains that are F plus and F minus, and therefore it is also known as the fertility plasmid. So the fertility plasmid, also known as the F plasmid, contains about 40 transfer genes which are arranged in a sequence one after the other on tandemly, and this allows for genes to be transferred from one bacteria to another through conjugation. So if you look into the function of fertility plasmid, they are involved in synthesis of pili, which is basically a physical contact between the two cells through which there is an exchange of genetic material. They're also involved in mating pair stabilization, prevention of mating between similar strains or similar donor cells, as also in DNA nicking and transfer during conjugation. Another important feature of the F plasmid is that they can function as episomes. So F plasmids are episomes, which are plasmids that can be inserted into the chromosomal DNA at times, right? And depending on whether the F plasmid is existing independently or is integrated into the host chromosomal DNA, you have another strain type that is called as HFR. As you can see, in this case, the F plasmid works as an episome and is integrated into the host chromosomal DNA. The episome can also excise from the host chromosomal DNA and again form a plasmid. So as the episomal form of the F plasmid excises, sometimes the excision is not perfect, and what can happen is that it excises with parts of the host chromosome along with it. So in such cases, it is called as the F prime plasmid, right? So F plasmids are episomes, which are plasmids that can be inserted into chromosomal DNA. The F plasmid are large, around 100 kb in length, and as I said, they have around 40 transfer genes. For the purpose of F plasmid to work as vector, they are engineered, and all transgenes in the F plasmid are removed. So BACs are engineered F plasmids without transgenes or transfer genes. So before moving into the main context of BACs, let's have a few revision questions. So here is your question. In blue-white screening, which parts of the LAGZ protein are coded by the plasmid and the host cell, respectively? Your options include A, alpha and omega, B, omega and alpha, C, both alpha and omega are encoded by the host, D, both alpha and omega are coded by the plasmid. So I'm sure you remember that the alpha fragment is what is encoded by the plasmid, and the omega fragment is what is encoded by the host, and the two come together to form a functional enzyme, right? So here you are. A is your correct option. Let's have another question. Name the enzyme that facilitates blue-white screening, and I'm sure you will remember that this enzyme is a part of the lacoperon, and your options include beta-galactoside permease, beta-galactosidase, and beta-galactoside transacetylase. And the correct option here, I'm sure you remember, is beta-galactosidase, right? So beta-galactosidase is the right answer. And just to give you a revision of the blue-white screening, so here you are. If you look at your blue-white selection, which is also known as alpha complementation, there are two types of transformants that you would have. One is the non-recombinant transformant, where the self-ligated plasmid would have gotten into the host cell. In the non-recombinant transformants, the plasmid self-ligates and therefore does not carry your DNA of interest within the LAGZ prime gene, which means that LAGZ prime gene is intact. And you have the recombinant transformants, where the plasmid carries your DNA of interest within the MCS, which is present within the LAGZ prime gene, right? So in case of non-recombinants, the LAGZ prime gene is intact and can code for the alpha fragment. So in both the non-recombinant and recombinant transformants, host cell is modified so as to only encode for the omega fragment of beta-galactosidase. So the Omega fragment is synthesized in both the transformants. The complete functional enzyme also requires the alpha fragment, and the alpha fragment is encoded in trans through the plasmid. So in case of non-recombinant transform, where the lag Z prime is intact, you have the formation of alpha fragments, and the two combine together to give you a functional beta galactosidase. However, in case of recombinant transformants, the gene of interest comes and sits within the lag Z prime locus, and therefore the lag Z prime locus is disrupted by the insert in recombinant plasmid, and therefore you do not get an alpha fragment. And hence, the functional beta galactosidase is not formed here. And how do you assay for presence of functional beta galactosidase? That is done by conversion of X-gal to blue color, which happens in the presence of IPTG, which serves as the inducer of the lacoperon. So in case of non-recombinant transformants, the X-gal is converted to blue color, and therefore the colonies are blue in color. In case of recombinant transformants, where there is no functional beta galactosidase, the X-gal is not converted to blue, and therefore these colonies are white in color. Right? So recombinants will be white in color, and non-recombinants will be blue in color. So let's now move on to describe the main features of a bacterial artificial chromosome. So here you have a typical bacterial artificial chromosome, and as you can see, there is one resistance marker here. For the purpose of selection of recombinants versus non-recombinants, you already have a lag Z prime locus, and you can go for blue-white selection. To ensure that the plasmid replicates, you have an origin of replication, and importantly, you have Rep-E, which is the protein required for replication. There are three additional genes here, which are known as PAR-A, PAR-B, and PAR-C, and these are basically responsible for ensuring the partition of the plasmid into the daughter cells. So as I said, these are engineered versions of F plasmid or fertility plasmid. The transfer genes have been removed completely. Anywhere between 150 kb to 300 kb fragments can be inserted into bacterial artificial chromosomes, but the average size is around 150 kb. They contain a selectable antibiotic resistance marker, which is known as chloramphenicol. And for selection of recombinants, you also have a lag Z prime locus, and you can go for blue-white selection to finally select your recombinants. A replication E protein that is responsible for replication, and the replication starts at origin of replication. PAR-A, PAR-B, and PAR-C genes regulate copy number and also the partitioning into the daughter cells. So as I mentioned earlier, in this case, we are not so much interested in amplification of the DNA as we are interested in maintaining a stable, large-sized DNA in the vector. So these are essentially low copy number plasmids. There is generally one copy per cell, and this is important because this prevents homologous recombination and ensures that the cloned fragment remains stable. Right. So now coming for the process of selection, the first level of selection of transformants is done based on the presence or absence of chloramphenicol resistance, which is because of the SCAMR gene. So if the BAC is present, whether it's self-ligated or recombinant, the cells would be resistant to chloramphenicol. The second level of selection for recombinants is done by lag Z prime here. You already know this is basically blue-white screening, and the lag Z prime locus here codes for the alpha fragment of beta galactosidase. When your DNA of interest is inserted here, then in that case, the lag Z prime locus is fragmented, and therefore a functional beta galactosidase is not found for the lack of alpha fragment, and hence the X-gal is not converted to blue color. Right. So therefore, the recombinants are white, and the non-recombinants are blue in color, where you have a functional beta galactosidase. And then, of course, you know that BACs are specifically designed for ensuring hierarchical shotgun sequencing of the human genome. So here you are. Transformants are selected based on the presence of chloramphenicol resistance or the CAMR gene, which would be present if the BAC has inserted into the host cell. If the BAC has not inserted into the host cell, then of course, you will not have chloramphenicol resistance. So those cells would not grow in a chloramphenicol plate. The lag Z gene harboring the multiple cloning site allows for blue-white screening of BACs containing inserts. The recombinant BACs would be white in color, while the non-recombinant BACs would be blue in color. And I've already mentioned BACS and YACS facilitated hierarchical shotgun sequencing strategy of the Human Genome Project. And again, though BACs are called as chromosomes, they are not true chromosomes for lack of centromere and telomeres. And in contrast, YACs are actually true chromosomes. So let's now talk about yeast artificial chromosomes, and in contrast to bacterial artificial chromosomes, these are actually true chromosomes. They contain a centromere, they also contain telomeres, and they have an yeast origin of replication. So these vectors contain elements typically present on chromosomes. They contain centromeres, telomeres, and an yeast origin of replication, and therefore these are true chromosomes, right? So the yeast artificial chromosomes are circular double-stranded molecules of around 11.4 kilobases in size, and they contain two selection markers for transformation. This includes TRP1 and URA3. TRP1 is involved in tryptophan biosynthesis, and URA3 is involved in uracil biosynthesis. And the selection process for transformants is basically what is known as auxotrophic selection. In this case, the host cell is deficient in synthesis of key metabolites, and these key metabolites can only be provided in trans via the vector. So the host cells that contain yeast artificial chromosomes are able to survive in minimal media that does not contain tryptophan and uracil, while the cells that do not undergo transformation are not able to survive. So basically, after transformation, the transformation mix is spread on a minimal media plate, and only the cells that contain the yeast artificial chromosome would survive, others would not survive, right? So that's the process of selection for transformants. The DNA of interest for the target gene is inserted into the SOP4 gene at a unique SNAP-E1 restriction site, right? And this leads to what is known as typically the red-white selection, which we'll discuss in details in the coming slides. And the important part of yeast artificial chromosomes is they can allow even larger fragments than bacterial artificial chromosomes to be inserted. The maximum size of insert that can be allowed in a YAC is around 500 kb or slightly more, right? So this allows for foreign genomic DNA up to 500 kb to be cloned into the yeast cell. So we now look at the actual process by which you make a recombinant yeast artificial chromosome vector. So here is your double-stranded circular yeast artificial chromosome vector. You do a digestion initially with BAMH1. You remove this fragment between the two telomeres, and then for the remaining vector that is there, you do a digestion with SOP4 to generate two fragments. You generate one arm that is from telomere 1 until the part of SOP4 gene here, and you generate a second arm that is the remaining part of SOP4 until the second telomere here. So this is how you do it. You digest with BAMH1 and with SNAP-E1. You generate two arms of these artificial chromosomes, one that contains the telomere, the TRP1 gene, the centromere, and part of your SOP4, and the second arm that contains the remaining part of SOP4, the URA3 gene, and the second telomere here, right? The DNA of interest is going to be inserted in this position here in the SOP4 gene, effectively disrupting your SOP4 gene, right? So next what you do is you do your ligation. You ligate blunt end insert here, and the insert comes and sits comfortably in between the two fragments of SOP4, generating a linear artificial chromosome that is a recombinant artificial chromosome. So this is more like a genuine eukaryotic chromosome in the sense it has a centromere which is intact, it has two telomeres, and then it has a certain number of genes in between, right? So this represents your disrupted SOP4 gene, and this is a linear recombinant yeast artificial chromosome. So while the yeast artificial chromosome initially is circular, after the addition of the DNA of interest, it becomes a linear chromosome, right? So the vector is cut with BAMH1 first to generate two linear fragments, and then with SNAP-E1 to disrupt the SOP4 gene. So this is your first part here. You generate two fragments of the vector by partitioning it at the SOP4 gene here, right? Then the DNA fragment to be cloned is ligated between the two arms, producing a linear artificial chromosome, which is this one here. This is your insert in the SOP4 gene, which is now disrupted. The ligated DNA is then transferred into auxotrophic URA3- and TRP- yeast mutants by protoplast fusion, and the transformants are selected by growing in these cells in minimal media. So as I told you, you go for selection of transformants via using an auxotrophic mutant host cell. These cells are deficient in synthesis of tryptophan and uracil. So these host cells are grown in minimal media, and only the cells that contain the vector therefore survive. The other cells that have not been transformed do not survive in this minimal media, and this allows for selection of transformants. And finally, the selection of recombinants occurs by what is known as red-white screening, which is based on the disruption of the SOP4 gene. If the SOP4 gene is disrupted, that is, it contains a gene of interest, then the colonies are red. If the SOP4 is not disrupted, that is, the transformant contains a self-ligated Yeast artificial chromosome, in which case you'll get white colonies. So now let's look at the mechanism of red-white selection. So red-white selection is basically linked to the process of synthesis of adenine, and there are several steps in this process. We'll not go into the details of these steps, but just to indicate, the starting molecule here is phosphoribosyl pyrophosphate, and then through a complex series of reactions, finally you have the release of adenine. One key metabolite in this process is what is known as p-ribosyl aminoimidazole, which is red in color, and this is then metabolized by ADE2 and ADE1 genes to finally give you adenine, which is colorless, right? So the mutations in ADE2 or ADE2 and ADE1 result in accumulation of the red metabolite, which is your p-ribosyl aminoimidazole, right? And this is responsible for the red color of the yeast cells in case they have mutations in ADE2 and ADE1. So now let's look at it in a more simple and representative fashion here. So this is your p-ribosyl aminoimidazole, which is coming from the pathway through ADE7. And if the host cell has non-mutated ADE2 and ADE1, there is a direct conversion of this red pigment into the adenine, resulting in colorless yeast cells. So in case of mutations in ADE2 or ADE1 or both, there is accumulation of the red pigment here, and that allows for the red color of the host cell. There is a second control mechanism by which mutations in ADE2 or ADE1 gene may be overcome, and this happens via the SOP4 gene. So in case you have a SOP4 gene that is intact and functional, in that case, mutations in either ADE2, ADE1 or both do not impact the final formation of adenine. Therefore, such host cells are white in color. So here you are, this is your non-recombinant vector here. The SOP4 gene is intact, and therefore this suppresses the mutations in ADE2 and ADE1 genes, which results in finally the formation of adenine, which is colorless. So such transformants that contain a non-recombinant yeast artificial chromosome are white in color. In contrast, if you look at the yeast artificial chromosome that is truly recombinant, so here you are, in which case your SOP4 gene is disrupted, there is no SOP4 gene product. Because there is no SOP4 gene product, there is no adenine formation, which means there is accumulation of p-ribosyl aminoimidazole, which is red in color, giving you red colored yeast cells. So this is what is typically your red-white selection. The reds represent the recombinants, the white represent the non-recombinants. So in case of recombinants, the SOP4 gene is disrupted, and this results in accumulation of red product, resulting in formation of red colonies, which are recombinant. White colonies are non-recombinant. Right. So let's now have a few revision questions before we summarize the lecture. So here you are, the first question: the genes involved in blue-white screening and red-white screening respectively are, and your options include A, beta-galactosidase and SOP4, B, SOP4 and beta-galactosidase, C, galactoside permease and SOP4, and D, SOP4 and galactoside permease. And I'm sure you'll get this right. So the correct option here is beta-galactosidase and SOP4, or the option A, right? Remember, beta-galactosidase is the gene responsible for blue-white selection, and the SOP4 gene is responsible for the red-white selection. Another important point of difference, in blue-white selection, the recombinants are white in color, but in case of red-white selection, the recombinants are red in color. So let's now take the next question. So here you are. Which of the following is the correct statement with respect to color-based screening of recombinants? So remember, we're talking of color-based screening of recombinants. We're talking both about blue-white screening as well as red-white screening. Option A, the recombinants are white in both blue-white screening and red-white screening. Option B, the non-recombinants are white in both blue-white screening and red-white screening. Option C, the non-recombinants are blue in blue-white screening and red in red-white screening. And option D, the recombinants are white in blue-white and red in red-white screenings. So if you see here, you have to really carefully read the expression so as to get what it is conveying. And if you pay attention now, of these, D is the correct option. The recombinants are white in blue-white screening and red in red-white screening, right? So the correct option is D.